So as Ethan said, my background is a systems person, but a number of years ago I went to a lecture very much like this one and got interested in the gene sequencing problem, and then from that led into doing actual core biocancer research, which is kind of what I do now. Which turns out to be really interesting, fun, and potentially very high leverage, so I encourage you all to think about it. So when I got an email saying, would you like to give a keynote at Sister, I said, I don't really do storage work anymore. And they said, as long as it's plausibly related to storage, come and give the talk. So this is the talk that you get. There we go. So this is a graph from IDC that shows 2 lines, right? The blue line is kind of how much total data is generated, which is not the same as what everybody would want to store, but how much is generated. And that does not include biological data, as you'll find out when you get later in the talk. And that's not actually the interesting part of the graph. The interesting part of the graph is the orange line, which shows how much total storage there is. And so IDC's estimate right now is that we've got 8 zettabytes, 8 times 10 to the 21st bytes, which is a very big number, but that'll come back. And I'm going to, in the end, show that if we used gene sequencing as part of ordinary medical care, and we did it worldwide, which is many enormous challenges to do that, but if cost was no issue of either the sequencing, the healthcare, or the storage, the amount of data that we could store is many, many, many times that. And so the bottom line for my talk is going to be, as long as we can reduce capacity, the cost of capacity, and also the sequencing people can keep pushing the cost of sequencing down, that this will drive demand for storage as long as you can produce storage. If we fail to keep the costs down, that will, at least in some sense, have adverse healthcare outcomes for everybody. So there's some motivation to do the work. Okay, so better part of 30 years ago, we had the Human Genome Project. The Human Genome Project took 13 years to do, cost $5 billion, and produced an amount of data that I think when they printed it out and put it on a bookshelf, that's not all of it, but it's almost all of it. And I think they printed it out on the bookshelf to show how big it was. Now, you're all storage people, and I'm sure you're not impressed with that amount of storage, right? I just stood up here and talked about zettabytes, and then I show you a bookshelf. What am I talking about? Well, then this happened. So this graph, this is a log scale with 5 orders of magnitude on it. This graph shows the cost of sequencing from the kind of end of the Human Genome Project to a few years ago. And the white line is if the cost had dropped at the Moore's Law rate of going down by half every 18 months. And you see that it beat Moore's Law by almost 3 orders of magnitude. Right? That is an impressive drop in costs. And the result of that is that instead of just being useful for research purposes, this is now useful for healthcare purposes, at least if we can get the biology right and understand what to do with the data, which is still an open question. Costs actually haven't dropped all that much since 2015, but I think that's more market than technology driven, in the sense that the company that makes the cheapest sequencers competitor can't beat their price, and so they have a big margin. So that can drop too, and probably will. Okay, so the talk that I'm going to give is basically in 4 sections. The first section, I'm going to give you some molecular biology background, stuff that maybe you would have learned in college if you weren't like me and I slept through my biology class. And so as punishment for that, now I'm doing it professionally. And then the second section of the talk, I'm going to explain how gene sequencing works in some detail. So that'll show you where the data is coming from. And also some of the pieces of it are very computationally intensive. So this is also going to drive demand for computation. For this storage conference, I don't really talk about that very much. The third part of the talk is basically a back of the envelope that I did that asks the question, if cost was no concern, how much sequencing data could healthcare generate? And I already told you the answer, right, which is more than an order of magnitude more than the total storage in the world. So there's some demand there. And then the fourth part of the talk is a diversion. It sort of flips the idea of the first part is that biological data is going to destroy your storage system by filling it. The fourth part talks about using DNA as storage and describes a project that's going on at Microsoft Research that I have nothing to do with, except I know the people to give me slides. But they're using DNA as a storage medium, so that'll destroy your storage system by completely replacing the bottom layer of it. And I think for this audience it's probably interesting because there are a lot of really good papers that could be written out of new properties of the storage system, so I'm going to explain that. And one disclaimer, none of the work that I'm going to describe in this is mine with the exception of the part 3 that's the back of the envelope hand wave. All the rest of it is other people's stuff, which is why it's good to get keynotes instead of your own work. Okay, so I thought I changed the slide. So in basics in biology, we're all made up of cells. Almost everything living on Earth anyway is made up of cells. Most of the work in cells are done by proteins, which are long strings of amino acids that fold into interesting shapes and do all kinds of stuff like transduce signals across cell membranes or catalyze metabolic reactions or bind onto DNA and cause it to get turned into more proteins or many, many, many other things. So basically all the interesting stuff in cells happens in, the first approximation happens in proteins. The sequence of amino acids that make up a protein are coded for in the DNA. So what DNA does in large part is just say, this protein that I'm going to make is the following list of amino acids. The process for doing that is the DNA gets transcribed into RNA, and RNA is chemically extremely similar to DNA, which you'll see in a couple slides. The RNA then gets exported from the nucleus, and a little biological thing called a ribosome runs along the RNA and makes protein, right? So that's how life works. All Earth life works that way. Space life, well, I want to see that paper. So there's this thing called the central dogma of molecular biology, which basically just talks about the information flow, and it says that data doesn't go from proteins up into DNA. The other thing that's interesting about DNA is that when cells divide, they copy the DNA, and I'm going to describe in some detail how that works because we hijacked that copying process to do all kinds of, like to do gene sequencing uses it, and the storage system really uses it. And so it gives rise to an interesting property in a storage system, which is that copy is cheaper than read followed by write, by many orders of magnitude. And I don't know of any other medium where that's true. And there's got to be a paper in that, right? You've got to be able to figure out something to do with that. Anyway, so that's basic biology. I'm going to describe probably in more detail than you care to hear what DNA is. So DNA is a polymer of nucleotides, and that little molecule over on the side is a nucleotide. It's made up of 3 pieces. The first piece that I just highlighted there is a sugar, right? So this is an oxygen and 5 carbons around it. That sugar is called ribose. And the DNA is an acronym. It means deoxyribonucleic acid. The ribo in the acronym is because of that. And the nucleic is because it's in a cell nucleus. In addition to the sugar, there's an inorganic phosphate. That's the thing that I highlighted. You'll notice there's a negatively charged oxygen on there. That negatively charged oxygen happened because it took one proton from a hydrogen that's in the water that's around it, which is the reason this is an acid. So the A in DNA is because of that negatively charged oxygen. And the sugar and the inorganic phosphate are how the backbone of DNA works. And then the third piece is the base. And so this piece changed, there's 4 different bases that you'll see in a minute. So the other parts of the DNA are always the same. So for reasons that I don't entirely understand, they number the carbons 1, 2, 3, 4, 5 in the carbon ring. And they call them 5' and 3', I don't know. But the way that the polymer in DNA works is that this hydroxyl here is the same as this hydroxyl there on the next nucleotide. And so the chain that runs through it goes it's got a direction from 5' to 3'. So strands of DNA are ordered. And the biological order is typically 5 to 3. So like the enzymes that work on DNA or the things that read it to make RNA go in that direction. Finally, RNA is the same as DNA, except it's got 1 extra oxygen there. So the D in DNA, deoxy, means it doesn't have that oxygen. So now you know what, if you learn nothing else, you learned what the acronym for DNA actually means. So the bases, there's 4 different bases: adenine, cytosine, thymine, and guanine. And conveniently for those of us in computer science, they all start with different letters. And so everybody just calls them A, C, T, and G. So if you look at this, this is a little bit more DNA. So one molecule of DNA is called a strand, and typically they're paired. So you see at the beginning you saw this double helix. Each of these strands is one half of the double helix. So the bases come in pairs between the strands. 8 goes with T, G goes with C. And if you were paying really careful attention, you'll see that those two strands go in opposite directions. The left one has 5 at the top, the right one has 3 at the top. So the two strands are called reverse complements from each other, right? Reverse because the direction's opposite, and complement because the bases are complementary. From an information theoretic standpoint, each strand has exactly the same information as the other one, so you can use one strand to make a copy of the other one, which is the basis of all life, so that's a kind of useful thing to know. And the other thing is, there's no actual preferred, like if you look at a chromosome, it's not like there's a forward strand and a backward strand. There's a convention by which we declared one to be the forward and one to be the backward, but the genes are about equally distributed in either direction. Like biology is random in evolution, and sometimes they get stuck in backwards and sometimes it gets stuck in forwards. And so the idea that one side of a chromosome is forward is just a convention, and it could easily have gone the other way. So the backbones of these strands are formed by covalent bonds that are pretty strong, and not just the backbone, but everything within the strand. Those solid lines are covalent bonds, while between the strands, the dotted lines in there that hold the two strands together are hydrogen bonds. And what's interesting about that is that they're about 10 times weaker than the covalent bonds. And so what that means is that you can add heat to the system to the point where the ambient thermal energy is enough that it'll break the hydrogen bonds, but not the covalent bonds, so it melts the two strands apart. This doesn't actually happen in biology. It needs to get up to about 90 C to do that. So when your cells divide, they don't actually heat up to 90. But it's really useful when you're, say, building a gene sequencer or storage system out of DNA. And you'll see that over and over again. So copying DNA, anytime a cell divides, it copies the DNA, and it tries to make an identical copy of the DNA. There's a protein, an enzyme called a helicase that pulls the two things apart, that does what you could do with heat. And then another enzyme called a polymerase runs down the two strands in opposite directions and synthesizes copies. And so now you have two double-stranded DNA when you had one. After it does the copying, there's what's called proofreading that fixes it up. It's got an error rate of about a little better than 10 to the minus 9, which if you tried to sell that on a computer would be terrible. But in a biological system, it's actually okay. And the truth is, if it was a whole lot lower than that, evolution would slow down, because that error rate is a lot of what drives evolution. And so from a species, organisms evolve as species, and from a species level, the errors in copying are actually helpful. So we were able to hijack, not me, but the scientists are able to hijack this process to copy DNA with a reaction called a polymerase chain reaction, or PCR. And you'll hear this again and again in the talk too. And it's almost the same as the biological version, except you use heat to melt the strands as opposed to a helicase. And one thing that's just a little cool factoid about this is that if you use, say, the human DNA polymerase, then when you use the heat to melt the strands apart, it would destroy the protein, because human proteins don't survive at 90 C. So they went off and found one of those sort of weird microbes that live near thermal vents at the bottom of the ocean that live at almost boiling. And those things evolved a DNA polymerase that can survive really high temperatures, because they live at really high temperatures. And so they use that to do this. So you don't have to add new enzyme for every cycle. And so you do one cycle of PCR, you double the number of copies of DNA, so you can get exponential growth out of it, which is tremendously useful for lots and lots of stuff. Okay, so away from DNA onto genomes. Human genome is made up of 22 autosomes, which are basically just chromosomes that you get almost identical copies from each parent, and then 2 sex chromosomes. So the ordinary thing is that you have 46 total chromosomes. There's 3.5 billion bases in like one half, like in one parent's contribution. So 6.4 billion total bases in a person. Only about 1% of that codes for protein, right? I told you that DNA has the instructions to make protein, but most of the DNA actually isn't protein coding. A little bit of it is sort of physical stuff. Like if you look at, these are chromosomes that are ready to divide, and they're attached to each other by this little piece in the middle. It's called a centromere. So it's like a physical spacer that's made out of bases of DNA. Similarly, the ends of chromosomes are these things called telomeres that are both a cancer defense and the fundamental mechanism of aging. I'm not going to talk about them again, but that's kind of cool. The remainder of the DNA, some of it is junk, some of it are endogenous retroviruses. They're just things that infected our ancestors somewhere, got stuck in our DNA, got inactivated, don't really do anything. But a lot of it are what called regulatory regions that are involved in deciding which genes get turned on when. And so even though they don't code for protein, they're really important and they have big health effects if they change. And so those things are interesting. The difference between any two humans with the same set of sex chromosomes is about 1 part in 1,000. So, we all look at each other and we look very different, but biologically we're nearly identical. And in fact, even the difference with a chimp is only 1%. So, chimps look really different from people, but they're actually not genetically all that different. And even yeast, which is about as far as you can get from people and still be a eukaryote, has got 31% of yeast genes have sort of equivalent genes in humans. So evolution has reused pieces a lot. Okay, so I'm going to switch gears and talk about something else because this is a place that we're going to get lots of data from. Microbiome. Everybody's covered with bacteria, and then your guts are full of bacteria. While with the cellular DNA that you have, all your cells have almost the same DNA if you don't have cancer. And almost everybody's DNA is almost the same. Microbiome is very different from person to person. It's different from part of the body to part of the body, and it varies over time. And it has large health effects on things, not just pathological microbes that cause infections, but non-pathological microbes affect all kinds of things of the way your body works and have very profound effects on your health outcomes. And it's not all that well understood yet. But the point for us is that there's a lot of bits of data involved in measuring the microbiome, so I'll get back to that in the third part of the talk. And then finally, cancer. So cancer is fundamentally a genetic disease. It's caused typically by mutations in DNA or by inherited bad things in DNA, that usually more than one, that combine to cause cells to grow without bound, and eventually they eat up all the resources in your body and can kill you. Cancer is a very widespread disease. About 1/3 of Americans will die from cancer. I was unable to find the exact equivalent number for Israelis, but it's lower. The overall cancer rate in Israelis is about 2/3 of what it is in Americans. But it's a very high number. And 15% of people around the world will die of cancer. You might think that the fact that the number is more than twice as much in the United States as it is in the rest of the world indicates that there's something bad going on with carcinogens. But what's actually happening is that people live longer in the United States or in the rest of the developed world. And cancer is mostly a disease of age, because very often it's accumulated damage over time, because you need to get multiple bad mutations in the same cell. So if you die when you're 40 of an infectious disease, then you don't get cancer when you're 70. So that's most of the difference in that effect. Unlike normal cells in the body, cancers very often have a whole family tree of cells. So that many cancers have the property that the proofreading part of DNA that I told you about before gets broken, and that causes the mutation rate in cancers to go sky high often. A consequence of that is that within a tumor, there's a whole family tree of different cells. So you've probably all heard cases or known people who show up with cancer, they go in for treatment, they get chemotherapy or radiation or something, they go into remission, they think, oh, I'm cured, I'm all happy, and then some number of months or years later, the same cancer comes back, it's resistant to treatment, and they have a bad outcome or they need a different treatment. Almost always what's going on with there is that there was some small group of cancer cells that was resistant to the treatment. And so you killed the bulk of the tumor, you feel better, you have too little disease to be noticeable, sometimes even too little to be measurable, and then the thing grows back out. That's important to us because you want to do a lot of sequencing of tumors to understand what's in them to treat them correctly in the first place, so this doesn't happen to people. Okay, that's the end of part 1. So we're gonna go to part 2 now, and I'm gonna tell you about gene sequencing. So gene sequencing, right, so I'm only gonna go through the sort of most common process for doing gene sequencing. There are each of these steps has many different pieces. There are different ways that you can do it. Different companies make machines. Lots and lots of people have written software, including this piece. I wrote some software with other people, not by myself. And I'm not going to describe my own work here at all. I'm going to describe the one that most people use. So typically the way that gene sequencing works is first you have a machine that takes the molecules and turns them into bits. And then you will take this, typically you get very short chunks of DNA. You get 100, 200 bases of DNA out of the 3 billion that's a person. And then so the second step is you take these little short chunks and you figure out where they best fit in this standard reference for the organism you're sequencing, so the human reference or the mouse reference or whatever. And then finally, you do variant calling, where you take all of the reads that align to one particular location and try to decide what's actually there. So I'm going to go through and describe all of these in detail. So the sequencer, sorry, I forgot the slide. So the kind of gene sequencing that I'm going to describe is reference-based gene sequencing. And what that means is that, remember I told you that the difference between people is only about 1 part in 1,000. So the Human Genome Project produced this generic human DNA reference, and it wasn't from one person, by the way. They took DNA from a bunch of different people. And so what you're really trying to do is to find the differences between that and the person or tumor that you're sequencing. So you start with something that's 99.9% correct and just try to find the differences. This point is important. There's way more data that comes out of the sequencing than you would think if you hadn't seen a talk like this. Typically, you can get hundreds of gigabytes to terabytes worth of data for a single sequencing run, and I'll explain why a little bit later. So first I'm going to start describing how one of the pieces of hardware works. This thing I'm going to describe is an Illumina sequencer. There are many other methods for doing that. Probably the most interesting of which is a nanopore sequencer that's actually a USB dongle. They're like that big. You plug them into your computer, you drop a little bit of DNA in and it reads them. But this is the most commonly used thing, and this is most likely what would be used in healthcare, at least if we did it now. So this is what I'm going to tell you about. So first you extract the DNA from the cell, you purify it, you split the DNA into little chunks. The size of the chunks is typically about 500 bases, but you can vary that, and I'll explain why later. So the basic Illumina setup has a chip on it, and the chip has got hundreds of thousands of individual cells on it. I'm only going to describe what happens in one of these cells, but you have to imagine that there's 10 to the 5 or 10 to the 6 of these going on in parallel. So each of these cells has got permanently bound to the cell a bunch of primers, and the red and green ones are reverse complements of each other, and those things are like stuck onto the glass that's at the bottom of the chip. So you take those 500 base chunks of DNA, you stick the same red and green primers on the end of it with a chemical process that sticks primers on. And then you put one in each cell. And so what will happen is one way this will either bind with the green end onto one of the red primers on the cell or upside down. I just picked a direction. So you do that, you have it attached to a cell on chip. You bend the thing over, and so now the green end of the DNA is attached to the red primer, the red end of the DNA is attached to the green primer, and then you run a single DNA polymerase, right? So you do that copying thing with the polymerase like you do in a PCR, but you just make one copy. So now you have double-stranded DNA. You melt that apart. Now you've doubled the amount of DNA you have in the cell, in the chip cell, not a biological cell. You repeat that until the cell has got lots and lots and lots of copies of the same DNA in there, or it's got reverse complements, right? So it's got the forward and the backward one. And then you wash off one direction. So you've only got, say, the forward side of the DNA. So now again, you've got 100,000 different cells with many copies of the same DNA in each cell. So here's the trick for how the sequencing works. I talked about DNA polymerase. What I didn't talk about is where the nucleotides that it adds come from, and they just sort of float in solution around and it finds them. So instead of doing an ordinary nucleotide, you do special nucleotides. So you put in an A, a T, a C, and a G, and each of them has got a little fluorescent molecule tagged on the side of it that are different colors. And then the other piece of it is, you remember there was it goes from 5' to 3', and on the 3' end there's a little hydroxyl that's used to attach the next nucleotide. So these special ones don't have that hydroxyl on them. They've got a methyl or something else stuck on there. I don't remember exactly, but they've got some chemical thing on them that prevents them from going farther. So you run a polymerase reaction, and you get one base added. So again, 100,000 way parallel. So each cell gets one base added, and the base has got a fluorescent tag that tells you what the which base it is. Okay, so now we're going to zoom out and look down on the chip. I think I have 36 cells instead of 100,000, but you take a picture, right? So you shine UV light on the thing so that it fluoresces. You take a picture, you get a color for each of those cells, and that color tells you what the first base is in each of the strands of DNA that you put on those cells, right? And then you go back, you remove that little methyl or whatever at the 3' end of the DNA. Okay, I lost a figure somewhere. That's what I get for editing the talk last night. But you remove a cap from the 3' end of the DNA, you repeat, so now you get the second base in it. You take another picture. Trust me, those colors are different than the ones I had before. And then you repeat that over and over again until you get 100 to 150 bases of that DNA, of each of those strands of DNA read out. And you can't go much farther than that because they start like blending into the nearby cells. I don't know the exact details for that, but that's essentially what you can do. So once you've got as far as you get, you duplicate the DNA. When we bend it over and duplicate it one more time and wash off the other direction. So now you have only the reverse complements in there from what you had before, and you repeat the thing. So now what you've done is you've read both. So you started with this 500 base pair chunk of DNA. You went off and you read one end of it, you flipped it over, you read the other end of it, and so what you have is two chunks of DNA that you read from each end of it, and then you have no idea what's in the middle. You can vary how far those things are apart by that 500 bases that you start in the middle. If you want to be farther apart, you do 1,000 bases. If you want closer together, you make them smaller. This turns out to be really useful because when you look in, say, the human genome reference, there are many places where you see the same sequence repeated, because that's the actual mechanism of evolution, is that things get copied and then they will sometimes mutate away from one another. So if you have just 100 bases, it's often ambiguous where it fits. But if you have some other DNA that you know that's 500 bases away, that may make this unambiguous. It's actually very, very helpful in trying to understand what's in some of these cells. Okay, so that's the hardware. The next piece of this is a computational problem. So this is called the alignment problem. This is the problem that I got interested in that started me down the biology path. So if you've got some read that's 100 bases, you got a reference that's 3 billion bases, try to find a place in the reference where the read minimizes the edit distance, which is the number of changes that it takes to turn the read into the reference. And you get differences from two sources. One is actual biological differences, and then the other one is that the Illumina process or any other sequencing process has got an error rate. Very often the error rate is 10 or 100 times bigger than the difference in, with Illumina it's about 10 times bigger than the signal. So you need to do this a lot. You could do this with brute force, but a typical sequencing run, you'll have a billion or 1.5 billion reads. So 1.5 billion times 3 billion with a nonlinear edit distance algorithm adds up to, you don't want to do that. Especially when you're doing it for everybody's annual physical. So I'm going to describe the tool that everybody uses to actually do this. It's a tool called BWA-mem, which I didn't write. So you take a read, you look at the beginning of the read for what's called a seed, so some number of bases, and BWA-mem uses a variable number of bases, but here we'll just assume it's a fixed number. It's got an index. It looks into the index. The index tells you the set of places in the reference genome where that seed occurs. So now you know those two places, the orange pieces in the read exactly match what's in the reference. So then you take the read and you sort of see how well it fits there, and you see how well it fits there, and then you go back and you pick a different seed and you repeat, you pick a different seed and you repeat, and after a while you get a whole set of possible locations that the read might align to, but you've done many fewer than 3 billion of them, right? You do a few hundred or fewer. And then you pick the best one. That's essentially how it works. The small advertisement for my software. So this thing is done with a Burroughs-Wheeler transform, which I'm not going to explain, but it's a wonderful data structure to build a compact index. Those things tend to be, for a human genome reference, it's I don't know, it's a gigabyte or something. It's really tiny. But it has the property that if you have a seed that's got length 20, every one of those 20 bases that you use is a cache miss, other than the first couple that are going to be in a cache. And so while this is great for space, it's sort of terrible for performance. The thing that I built uses a 30 gigabyte hash table that does one cache miss, which is part of the reason it's 20 times faster than this. Okay, so enough advertising for my software. So the output of these two steps are a file that's in a format called SAM or BAM. They're the same data. SAM is a text file, BAM is a compressed, it's basically just a gzipped SAM file, except you gzip every 64K so that you can seek into the middle of it, but that's really all it is. But I'm going to describe what's in this in a little bit of detail because this is what's actually going to occupy all the world's storage. So it seems worth seeing one line. So this is most of a sample read line from a SAM file. Like the first place is just a unique ID. It's just a random string, basically. There's some flags that tell you stuff like how the alignment worked and whether it's a duplicate or things. That tells you the chromosome and location that aligned to. There's an estimate that the aligner said of how likely it thinks it is to be right, which usually means how many places that I find that we're close to as good as a match of this. The cigar string describes where the differences between this and the reference that you align to are. And then if it's a paired end, like the Illumina kind where there's two reads that go in a pair, it tells you where its mate pair is. And then where most of the data winds up are in these last two fields. So the first one is the actual sequence data that came off of the sequencer. So I only show a few bytes of it there, but that's actually 150 bytes. And then the last one is the base call quality. And so remember, the Illumina machine takes a picture, and the picture looks at colors, and the colors are not always perfect. And sometimes you can imagine a cell where the polymerase didn't work exactly right, and so you actually have more than one fluorescent tag in the same cell because it's not perfect, and there's no proofreading in there. So so you get all kinds of things that can cause errors, and that gives you an estimate of how likely the bases are to be correct, and those are actually really important in understanding how to deal with the read. So you might look at this and say, okay, we've got a bunch of little fields and two big ones. This thing is probably only different than the reference by 10% maybe, because that's about the error rate, or a few percent. So can't we just compress this by storing it as a delta from the reference? And it turns out you can, and there's a format called CRAM that does that. But these guys only had 2 bits of entropy anyway, because there's only 4 bases. So it's an alphabet of size 4, and gzip is like really good at Huffman coding, and it uses 2 bits to store this. And these guys have more like 8 bits of entropy, and they're not relative to the reference. So all the data is in that. Then that means that it's hard to compress beyond sort of basic gzip level compression. Okay, so the third phase of gene sequencing is variant calling. So you have all these reads. They came in some completely random order because it depends on how the DNA got deposited on the chip. You align them, and then you sort them, which now we're sorting like 1.5 billion things, so the sort is tricky. And then you say, okay, well, I'm going to look at all of the reads that go to a certain place in the genome. You can't just have one, because if you had one, and they say that like the red thing is a difference between what was in the reference, that could mean that that person had that there. It could mean that there's an error in the sequencer. It could mean that there's an error in the aligner. You just don't know. And furthermore, for most of your DNA, you've got 2 copies, one from each parent. And so it could be that one parent is red and the other one is a blue or green or whatever, and you can't tell. So what happens is you have what's called sequencing depth. So you'll arrange to have many more than one read aligned there. And the reason you need to do that is so that you can like wipe out the stochastic error from the sequencer, and then you can tell the difference between what's called heterozygous locations where one parent has one and one parent has the other. And so a typical depth that you need is overall coverage of about 30 times. So you need about 30 times as many bases and reads as there is in the reference genome. The third piece that causes you to need that much is that they're not distributed uniformly, so there's some stochastic variation in depth, and you need to have enough depth to disambiguate what happens at the places with the lowest coverage. You'll see much higher depths for cancer that I'll explain later, but just for a normal one, you need about 30 times as much as the genome. And so that's the reason that these files are so big, because you need lots and lots and lots of reads, and then there's a lot of entropy in each read beyond just the genetic part of it. Okay, so part 3 of the talk. How much sequencing is there going to be? So we go back to the graph that we saw at the beginning, and again, this is the interesting part. We've got 8 zettabytes of storage to work with in the world right now, and you can see kind of the slope of the curve. It's going up, so maybe we'll get to 10 zettabytes next year, but it's not going up that fast. So first question is, imagine we just took everyone and sequenced them, right? That seems like a really useful thing to do. Your healthcare outcomes depend in many, many ways on what's in your genome. It seems like a thing that should be in your medical records, right? So that, I mean, it seems like almost like a no-brainer if you ignore the privacy implications of this, which, you know, we already have plenty of personal things in medical records and know how to deal with that, sort of. So if you do a 30 times coverage whole genome set, it's about 100, that's about 100 gigabytes to sequence everybody. So, all right, you generate 100 gigabytes per person. The world population is 7.4 billion people. You multiply that out, and you get 740 exabytes, right? So that's like 1% of the world's total storage capacity. So that's not trivial. It's not cat videos, but it's not trivial. You know, but I told you at the beginning of the talk that we're going to blow away the world's storage capacity, and this isn't doing it. So something else must be going on. And a big chunk of what's going on is cancer. So with cancer, because of that problem where you have a whole family tree of different genomes inside a tumor, for not all, but for many, many tumors, and the fact that the small pieces of the small subpopulations of that greatly affect treatment outcomes and whether people live or die, you can't, or you should, if you had your choice, you wouldn't sequence a tumor to a depth of 30. Instead, you probably sequence it more to like a depth of 1,000. Now you get a little bit better compression because there's a lot of duplicate reads when you do a depth of 1,000. So it's still several terabytes per run. Okay, so we do several terabytes per run. About 40% of everybody in the world will get cancer at some time during their life. That's a scary number, right? But many of us will wind up with cancer. Probably some of us have it already and don't know. Hope not, but we, you know, the odds are not good for all of us to be cancer-free. So if 40% of the world's population get cancer, then that means that we have 3 billion tumors that will happen in the current world population. Of course, the population is going up, so these numbers will all go up over time. And furthermore, tumors evolve. They evolve a lot when you treat them because you provide selective pressure, but they also evolve when you don't treat them because the cancer genomes are changing around to try to find more fitness, right? Fitness for tumor is bad for patient. But when the cells get better at growing faster and faster, you see evolution even in untreated tumors. And so if you're an oncologist, you probably want to see what's happening in the tumor, say, monthly. Now, this is expensive. Nobody actually does this now. But this is something that you would like to be able to do if cost wasn't an object, both of the sequencing and of the storage, and then getting everybody to come to the oncologist when they have cancer, which on a world scale is also an incredibly challenging problem, but not our problem. So you multiply all those things out, and you get 3 times 10 to the 23rd bytes, 300 zettabytes. Well, okay, remember I told you we had 8 for storage, so we've got 38 times the world's storage capacity to store the sequencing data for everybody's tumors. So we have some work to do, right? I mean, and you probably want to keep this around because remember, tumors go away and then they come back. This isn't data that you want to throw away. In real life, because cost is an object, you wouldn't actually store all of this data. You'd store summaries of the data. You're clearly not going to do this for everybody because not the whole world gets the best possible cancer treatment. But aspirationally, this is where we'd like to be. So what this is really saying is that as you build your storage systems, you have to imagine that demand for cold storage capacity has at least 2 orders of magnitude more than we currently have, right? So as we keep reducing the cost of storage, this, among many other, and I think it's a better thing to do with it than surveillance videos, right? I mean, all in all, it's probably nice to be able to go back and see who was on what corner in London at some particular time 5 years ago, but curing mom's cancer is better. Okay, so last thing that might generate an enormous amount of data is microbiome. So remember, I told you about the microbiome. The 10 to the 3 number I made up. So there are 10 to the 4 possible different microbes that can live in people, but there's an enormous difference from person to person, from region of the world, based on diet, who you interact with. So if we conservatively assume that only 10% of the possible microbes live in any given person, so you've got 1,000 different types of microbes. The genome of most of these are bacteria, not all are bacteria, but most of them are bacteria. Bacteria have genomes that are much smaller than people, so it's like 1 million bases as opposed to 1 billion. So that's 3 orders of magnitude smaller. Oh, actually, I forgot to say this. So there's this trope that's been going around for a while that came out of one paper that says if you look at a human body, 90% of the cells are microbial cells or bacterial cells. That's actually a subject of debate in the research community, and the number is somewhere between 1 to 1 and 10 to 1 in favor of the microbes. But even if it's 1 to 1, you got a lot of microbes. And contrary to what actually a famous computer science professor who you probably all know, whose name I won't repeat, said on Facebook, getting rid of the microbes does not mean that you lose 90% of your body weight because they're really small. She was interested, she was making a joke, but wrong button. So if you multiply those out, you've got about 1 billion bases of microbial genome per person, but with a human genome, they're all the same, or if they're in a tumor, they're not all the same. With this, you've got a large variety of them. You also have the subpopulation problem in microbes, right? Some microbes are very common, some of them are not very common. For instance, there's a bacterium called Clostridium difficile that's a gut bacterium that's present in almost everybody. And very often when you treat someone with antibiotics, what will happen is it kills the other bacteria in your gut, but C. diff is antibiotic resistant. And so the surrounding flora in your gut go away, the C. diff grows out. It turns out to be, if you have too much of it, it's very bad for you, and people die from this. For a long time, the treatment for a C. diff was to give more antibiotics, which didn't work it worked a little bit. Turns out that the way they treat it now is with stool transplants, which is gross to think about, but really it turns out to be remarkably effective. It almost cures everybody on the first treatment, and if it doesn't on the first one, the second one pretty much gets everybody, and it doesn't make you sick like high-dose antibiotics. But understanding that kind of stuff is in your gut can have big health impacts, and so you really want to know. So if you multiply that together, you probably wind up with a scan that's maybe not quite as big as a cancer scan, but say 10 times bigger than the human genome scan. And this seems like an annual physical kind of a thing. So if you do that times the world population, you get 7.4 times 10 to the 21st bytes per year, which is about 90% of the current world's installed storage capacity annually, just for the annual physical data. So again, there's a lot of demand here. In real life, it won't materialize because cost is an object, and being able to deliver healthcare to the third world at the level of doing genome scans of their microbiome every year is a challenging political problem or economic problem. But if we could, then the demand for the storage are there. The other disclaimer is this is really back of the envelope-y, so you can quote me on it, but you have to include the disclaimer. Get that right. And you could argue about the utility of some of this data, but I think while in real life you would probably store summaries to save money, that will, at least in some cases, compromise healthcare outcomes. So more bytes or more grandma stays alive. Okay, so now we're going to shift gears completely and go into the final section of the talk, and we're going to talk about using DNA to store data as opposed to using DNA as a source of data. So this is a project that's called PALIX that's running jointly between Microsoft Research and the University of Washington. The PIs are married to each other, which I think is good. They seem to like working together. And these are all Karen's slides. So if you look at DNA, right, we already know there's 4 different bases of DNA. So essentially what that means is that in principle, you can get a density of 2 bits per base of DNA that you store. In real life, it's orders of magnitude worse than this because of practical things, but the DNA is tiny. If you remember way back at the beginning of the talk, I had pictures of human chromosomes, and there's a scale on the bottom of that, right? And the chromosomes are about 5 microns. And the biggest of the human chromosomes has got 250,000 bases in it, right? So you can store, 250,000 bases times 2 bits is like, what is it? It's hundreds of megabytes in a 5 micron line, right? It's not 3-dimensional, it's a very tiny thing. So there's a lot of density here. So the basic idea is that we're gonna make synthetic DNA. We're gonna make short strands of them. We're gonna store it and then read it back. So the density is insane. So this is a picture of data center. Karen's estimate is this maybe an exabyte of storage in the data center, right? So it's a 1/100% of the world's storage capacity in one of these giant things, versus storage in DNA. And you probably didn't notice it, but there's a dot there. Now, this is not actually, to be completely fair, this is not an apples to apples comparison, because the data center has got things like power and cooling and networks and empty space, and this is just the DNA and none of the hardware. But it gives you an idea of what we're talking about here. So here's a little slide that she did that shows the potential storage density of a bunch of different things. And again, this is a log scale graph, and these are orders of magnitude. So this is a very, very, very big range, right? So we think of disks as being pretty dense, but their density is way down here. The blue chunk is estimates about how good it can be, which means wherever she got this from, saying the density might go up by an order of magnitude over some number of years, which that would be 100 terabyte size disks, which maybe we'll make. Right, SSDs are of course not going to get that big. Flash, when you measure just the chips, has got higher density because it's all chip. Tape is kind of the cold storage that we've got now. And tape is like pretty good. It's several orders of magnitude better than disk because, again, you don't store the whole mechanism with it. You can just store the medium. Right, this is holographic storage. That's all future, right? It's not a deployed thing. And then there's 7 orders of magnitude between projected future for tape and DNA. Now, in practice, you're using many orders of magnitude of DNA for things like coding and redundancy, because you can't, any individual strand of DNA has got a pretty decent chance of breaking, so you want to have hundreds of copies of everything that you've got in there. But even when you spend orders of magnitude for metadata and spacing and redundancy, you're still 3 or 4 orders of magnitude denser than tape, right? That's a crazy, crazy number. So they've actually built this. They got a Nature Biotechnology paper. They're not storing exabytes of data. They stored a gigabyte of data. It's like, I think it's an OKGo video and a copy of the Bible and some other. I think OKGo was cool with it because they're cool. But so they stored some data. They wrote it into DNA. They read it back. They demonstrated that it can be done. They're, I think, like an order of magnitude more data than anybody's ever written into DNA. And there's lots of reason to believe that you can scale this up over time. So another really interesting property of DNA that's unlike any other storage medium is it's actually got demonstrated storage longevity that's insane. So people find woolly mammoths that were buried in the last ice age. The glaciers melt back, the mammoth comes out, you can sequence the mammoth and find out what's in it, right? So that's 10,000-year-old DNA, essentially completely intact. You can go in fossils, and she says 2 million years. I'm not sure you can get it that long, but at least 100,000 years you can get a decent amount of DNA. And the mammoth was frozen. This thing was just sitting somewhere. You can still get bits out of that. So this literally has better demonstrated longevity than carving stuff in stone, right? You carve stuff in stone and it weathers away over centuries. This doesn't, and it's got way better density than carving in stone. So, when you compare to the lifetime of all the other media that's years or a decade, you're better by orders of magnitude. So if you want to save stuff forever, that's one reason to do it. Another reason to do it is that with other, over spans, when we're talking about 10,000 years, societies change, technologies change, and it may be that nobody knows how to build a reader for your holographic storage thing anymore. Well, DNA, the coding isn't, but the basic storage medium is built into everybody's biology. So even if we had some kind of horrible apocalypse and then society rose back up again, they're going to figure out how to sequence DNA for the same reason that we figured out how to sequence DNA in the first place. And so then the problem in extracting your storage is just finding the physical storage and then understanding erasure coding, which is much simpler than completely understanding a new medium that may have rotted away anyway. So there's again, if you really, really want to save something, this is a great way to do it. The other thing that's amazing about this, and that I think should lead to some interesting submissions to SysStore or hot storage in the future, is that we have a medium where copying is cheaper than read followed by write, because you can just run PCR. And another really interesting property of this is that because PCR only amplifies things where primers match, you can effectively do chemical seeking in DNA. So you can go in and say, if you've got some bases that are at the beginning of the DNA, and you've got to choose what's there because you designed the format, you can selectively amplify those and then read them out while not reading everything else that's in the little droplet that you've got. And again, there's got to be a file system paper in that. And I would love to read it or review it in the future. So, this is the audience that might care about that. So we have that. So basically, if you imagine one of these DNA storage systems, the fundamental thing, you start with some bytes, you do some encoding. The Palix guys have worked out the encoding, and there's some real challenges in that because you're writing into sort of 200-bit strings, and you've got to do addressing and redundancy and error correction in 200-bit strings. But we have some mathematicians. I looked at their work. It seems very good. You synthesize that into DNA and preserve it. They're attaching it to these little glass beads and putting it in little tiny tubes. And then you can access it randomly both at the tube level and then also within the tube by doing the chemical seeking, the selective PCR. So you can pull it out. You can use a gene sequencer to read it, reverse the encoding, and you get your bytes back. So in principle, you build something like this. And if I think in with this very robotic-looking person there, I think this is probably enough to store more than the total world's storage capacity now in something the size of a person. So, that's pretty useful. So in principle, you build something like that. In practice right now, what we have is graduate students with pipettes. So, it's not quite the volume there. That's one of the co-PIs that's standing looking over her shoulder like he doesn't do it anytime when they're not taking a picture. So this is the real end-to-end system that they built. So the left-hand side of it does synthesis, the middle is storage, and the right-hand side is sequencing. This is actually, you can't see the sequencer because there's a thing behind it, but it's one of those nanocore sequencers that's a USB dongle. They did a bunch of work, like this is really cool microfluidics that lets them move little droplets around to store and sequence them that I don't have the video of here. So that's DNA storage. I'll conclude the talk now. There have been tremendous advances in biotechnology that made it practical to measure all kinds of stuff about bodies. I only talked about DNA. You can imagine measuring many, many other things, and earlier drafts of the talk that ran in 2 hours had this in it. But you can imagine measuring proteins, neurons, all kinds of stuff. There's going to be lots and lots and lots and lots of medical data that might be valuable to people's healthcare. We won't really store them all because storage and measurement takes money, but as we reduce the costs, the demand will go for at least 2 more orders of magnitude from what we've got now. Eventually, we'll need yottabytes of storage. And like this, maybe unlike, it won't be the astronomers, it'll be the biologists and computer scientists that force them to come up with the next metric prefix, because 10 to the 24 is the currently biggest named metric prefix, and we might need 10 to the 27. So that would be kind of cool. And one way that we could do is we could store DNA in DNA. It's got great density, got great longevity, and the format will be around forever. So that's it. This is a picture of a gene sequencer, by the way. So it's like a tabletop. The big gene sequencers are tabletop things, and the little ones are just dongles. So I will take questions. Yeah.
Read-write speed.
Oh, read-write speed. So the read speed is pretty good. It's like megabits because it's driven by it's driven by sequencing technology. And of course, you can make it as parallel as you want. Write speed is lower and proprietary, so I can't tell you, but they're working on increasing the write speed. The latencies, however, are terrible, right? I mean, the latencies are not milliseconds, they're minutes. So think of this as competing with Glacier, like with Amazon's Glacier, not with an SSD where you talk about latencies in microseconds or 3D crosspoint where it's nanoseconds. This is minutes.
It's like burning a CD-ROM or something.
Yeah, it's a lot like burning a CD-ROM, and the write latencies will be even bigger. But for that, you probably don't care. I think we'll probably put a cache in front of it. I'm guessing, but probably a cache. Yeah.
Related to the previous question, it seems like an enormous overkill on capacity, density, at the price of other parameters that we just mentioned. So, because I don't really care if the whole world's storage is this big or 1 billion big.
Mm-hmm.
Either is definitely okay. So the question is whether the right thing to do or a better thing to do is not to look for something that's a few orders of magnitude worse in density, but much more convenient in access speeds and things like that.
It may well be. It may
I
well be.
understand Microsoft has a glass storage project. Where does that sit in the? You mentioned several technologies. I don't think that was one of them.
It was on there, but I didn't. I barely talked about it. I believe that was optical is the glass storage. I don't know much about it, so I can't really comment intelligently. In terms of the basic question, in terms of the basic question of would you want storage systems below the parameters? Yeah, of course, right? Like everything else that happens in hardware, this has got pluses and minuses. And the question for us is, can we do something useful with what the technology provides? And the answer might be no, but anything that differs by that many orders of magnitude is at least a fertile place to write papers about. And so, we'll try it and see. Yeah.
So I have a follow-up question, because what is interesting to me is how big can a reader become, or how small can it become, and how small can a writing entity become? And have you ever made the on the envelope calculation how much space you need to get to a certain throughput?
I believe Karen has made that calculation. I may have even seen the slides, but I don't want to quote them because I don't remember them very well. Readers can get very small, right? I mean, like I said, the current, not this kind of reader, but the nanopore readers are like that big.
Chip size. You're shown them.
Yeah, and they, and honestly, getting it much smaller than that, you have to pipette the DNA in. So if it's smaller than that, it's hard to work with by hand. Now you can imagine like microfluidics doing it or something when you'd scale it out. But and they cost like $1,000, right? You can buy one and plug it into your PC for $1,000. So you can get the readers to be small. Much less effort has been put into writing technology, because reading technology is useful for biological and research applications and now medical applications. And writing has got some biological applications and computer storage applications, but it's all very new. And so, it's many orders of magnitude less effort. So, Ethan. Yeah, Ethan.
So given following up on that, given that statement, one of the points you made is, and it was on the slide, I don't know if you mentioned it, is, well, gee, we're doing all this work to read someone's DNA, decode it, only to turn it back into DNA in a somewhat less efficient way. It seems like if this is what you're going to be doing, you could just take the DNA
and store it
and say, hey, we want to read it again. Well, we can read it very quickly. Yeah. We've already said that. Yeah. Just read it again.
You could do that. That's a little bit harder to do with the microbiome stuff because it's a bunch of DNA collected from a bunch of locations. But yes, you might imagine. You might imagine storing the DNA rather than
right. And I'm just looking at it that way and say, well, look, if you're going to want to sample the cancer tumor every 36 times over the lifetime of the tumor, well, just 36 slots and store them. And if you ever need them again, resequence them, because that way you can leverage the reading, which is much better right now, and don't worry about the writing.
Yeah. Well, and to be clear, I wasn't saying that we're going to store all this in DNA, right? We can store it in disks. Yeah. We can get 2 orders of magnitude of storage out of disks and tapes. Mm-hmm. And we will. I'm sure we will. But yes, storing biological samples is one thing that you can imagine doing. Gala.
So you talked about capacity and cold storage, but how is the biological data used? What's the access pattern when it's hot on
The access patterns tend to be, depends on what analysis you're running on them. They're either whole file sequential or small pieces random access, depending on what you're doing. But it's not like whole file random access. So you're either going to read a few 64K chunks spread out all over the thing to find some particular pieces of the genome, or else you're going to reprocess the whole file and then you just read sequentially. So it's actually very convenient storage system wise. Well, yeah, somebody,
Go ahead.
Michael.
Have you thought about, or others thought about, if I have the sequencing for 7.4 billion people, and I find something interesting in one person's sequence,
Yep.
how do I find and index the data such that I find the 17 other people out of those 7.4 billion that have that? same
Yes,
interesting thing?
you would want to build an index, and that would occupy a lot of space. And no, I haven't worked on that problem. There's interesting ethical problems too that you need to overcome about whether you want to use people's healthcare data to do research. But I'll assume that we solved those because that's actually probably easier than sequencing everybody on the planet. It's a legal problem, and having been in the gene sequencing community, a disturbing fraction of them are lawyers worrying about consent because the medical folks got really burned by the Henrietta Lacks thing. They I don't know if you know the story, I'm not going to really tell it, but they used biological samples from a cancer patient without her knowledge or consent and then published them with her name, and her family objected strenuously to this when they found out years later, and to their credit, they learned the lesson not to do that again. And so now they're very concerned about informed consent about everything. So there's a big legal problem. But yeah, you would want to build an index, and that's an interesting question, how you build an index of 7.4 billion genomes. And it's probably not much harder than indexing the web.
Yeah,
So.
it may be easier because you can leverage some of the behavior of DNA with respect to one another. So you may be able to do it I'm not saying I know how, there may be chemical ways of putting a whole bunch of stuff in a thing and having the ones that are similar.
Oh yeah, you could do that.
I'm not saying I know
You could do
how,
it
but it
chemically.
seems like you ought to be able to.
Yep.
Maybe you mentioned, is it cheaper? Is there something if I have two samples of the same tumor or not? Is it cheaper to sequence incrementally? In other words, is there anything gain from sequencing the same but slightly mutated?
Not really. I mean, so there are minimum batch sizes for sequencing. So if you have a very small amount of stuff that you want to sequence, then you want to batch it together with other people. And there's actually technology for doing that where you can stick barcodes on the DNA. They're DNA barcodes, but they call them barcodes. But when you're sequencing something as large as a tumor, you're way past the batch size, and there's no advantage to doing that.
Questions that is connected to atoms. So just to understand the potential, you said that DNA, so DNA looks complicated. It's a complicated thing.
Mm-hmm.
Why can't we use something that is more simple? I'm talking about the last part of storing.
Oh,
So why not storing in a much simpler with less atoms molecular than DNA? I mean, the tooling is there, so that's a very good thing. But the only thing you're using is the PCA process or the copying process. But other than that, you can use whatever other.
You could imagine doing that, and you already answered your own question. The copying piece is really important to make this work because in practice you need to store hundreds or thousands of copies of the data because not all the molecules survive or you can find them. And the ability to design a copying enzyme is far, far beyond what biologists can do. Like we're nowhere close to being. Nature made these and we just copied them from microbes. But
But from density point of view,
from a
the amount
density point of
atoms
view,
that you have in the DNA,
you
is-
could design, if you just wanted to do storage, you could probably produce something that's more dense.
exactly.
But
Yeah, but that's already more dense.
But then you can't leverage gene sequencing hardware, you can't leverage copying enzymes. And, as Ethan pointed out, maybe we already have enough density with, somebody pointed out, we have plenty of density already.
There are people looking at this problem. I think it's Marie Curie, France. I forget the guy, Jean-Francois something.
There are people looking at using proteins for data storage. There are people using synthetic genomes that have 8 bases instead of 4.
Right, yeah, and you can do all of that. Proteins are probably more dense because they're 20 amino acids
Right, so
instead
you have
of
to be able to
core.
read them and write them.
And
That's the
reading
question already
proteins
answered.
is much harder than reading DNA. They do it with mass spectrometry, and it's actually really tricky to read proteins compared to DNA. It may not stay that way, but right now you can't just run it through like a nanopore sequencer and read it. Yeah.
Last question.
Last question.
And the durability of the data stored there, how long it lasts the data?
Tens of thousands to millions of years. I mean, if you can keep it cold, it'll probably last a million years. So longer than you care. far longer than any of us will be alive.
Longer than glass?
Unless you can take a relativistic space journey, we'll all be dead before it. You're not going to line. I mean, the way that you lose the data is you lose the data center, not the grades. Okay, thank you very much.
Thank you.
Thank you.